Okay, now we're going to talk about the chromosomal basis of inheritance. And what this means is basically how do we get some of these non-Mendelian inheritance rules that we see in a lot of things. So again, let's review Mendel and Mendel's rule of independent assortment. Remember, he looked at 7 different traits in peas that were all on different chromosomes. And he didn't know anything about genes or chromosomes. He just knew these factors tended to segregate and recombine in predictable ways. And today we know that's because these genes are on chromosomes, and the chromosomes segregate during meiosis and produce sex cells that then recombine during fertilization. So that Punnett square that you do really represents the segregating of alleles, which you put on the top and side of the Punnett square, and then the recombining of alleles in the boxes of the Punnett square represent fertilization. So for example, if we looked at the color and the texture of seeds, right, yellow versus green, round versus wrinkled, he got this, if he crossed a yellow round, he got all showing both dominance, right, in the first generation in the F1. In the F2, he got this 9:3 to 3:1 ratio. He found that there was this segregation of each of those pairs of alleles and recombining, which he termed independent assortment. Okay, well, again, Mendel published his rules, these rules and his heredity factors, and then other scientists followed up and experimented with them, but they didn't always use pea plants. So for example, another organism that's useful to use is a fruit fly, Drosophila. And a guy named Morgan in the early 20th century looked at Drosophila, and what he found were sometimes he got these non-Mendelian conditions that were occurring. For example, when he crossed, he found a variation in eye color that produced white eyes, and he found that that almost always appeared in males and only in males. And he looked at that and said, if Mendel's laws are true, that both sexes should be affected equally, right? And he came up with this idea of sex-linked inheritance, and that we must have a chromosome that's unique to males versus females, right? And that if you have a recessive allele on your X chromosome, females have 2 of those, males have 1, that you will get these different ratios in expressing a recessive allele on the X, since a male can only have 1. So this is an example of how that sex-linked trait would be passed on in these fruit flies. So let's look at this in a little more detail. So if you crossed a female, let's say she showed the dominant condition, in this case red eyes, well, all the offspring are going to have red eyes. So that's no big deal. That shows simple Mendelian dominance, right? However, if you have a female and she's heterozygous, she's going to pass on that recessive white-eyed allele half the time. If you cross her to a male with red eyes, right, all of the females will have red eyes, but half of the males will show that white-eyed allele. Okay, so again, we're seeing a difference between males versus females. The only way you can get a female showing that recessive is that the father has to have white eyes, and the mother, the female, must be a carrier, and then you can get a female that produces those white eyes. But again, most of these recessive alleles are kind of rare, so if it's maybe 1 in 50 or 1 in 100 individuals have it, it's very rare that both parents would carry that recessive allele. So you very rarely see these sex-linked traits in females because they have to have it on both X's. So again, what would that look like in humans? Something like hemophilia. Well, if a mother carried hemophilia, which of her children could have hemophilia or could carry the gene for hemophilia? Well, again, her X is going to go to both her sons and daughters, okay? But the hemophilia one is going to go to half of them. So that means that 1 of 2 sons, or half of her male sons, would have hemophilia, and half of her daughters would get that X, but they'd get the normal X from dad, so they'd be carriers. They would carry that allele. They would not have hemophilia, okay? How about if dad has hemophilia? Is that a concern for the kids? Are they going to have hemophilia? Well, again, dad's going to pass the X on to his daughters and the Y onto his sons. Both males and female children will get the X from their mother, so none of the children will have hemophilia, though both of the daughters will carry the allele because they have to get the X from dad. If they get dad's Y, they're not daughters, they're sons, right? Okay, so again, a little more on sex linkage producing these sort of non-Mendelian inheritance. And then tortoiseshell cats, we talked about this a little bit in class, how you can have a codominant condition like orange and black fur, that's because it's on the X, and X inactivation means that in any given cell, one of the X's is turned on, one is turned off. So if you're heterozygous and you have a black and an orange allele, in some of them you'll have the orange allele expressed, I'm sorry, that one, and then others you'll have the black allele expressed. Again, what we call codominance, another non-Mendelian inheritance pattern. Okay. However, if we look at males, right, so females can have both. Males, since they only have one X, will either have the black allele expressed or the orange allele. So again, another factor where we see, because of these conditions on the X chromosome, we see differences in patterns in males versus females. Okay, so Morgan also looked at a lot of other traits in fruit flies. For example, he looked at coloration, black color, which is little b, versus brown, wild color, which he just called B+. He put a positive on there for wild type. And then he also found some that had vestigial wings, little shriveled up wings that don't work, versus normal wings, which again, the wild type is plus. So when he crossed two of these individuals, obviously, if you cross a homozygous for the dominant wild type to these recessive conditions, you get all individuals that show the dominant. So that's what this individual was. This one is heterozygous. It's got expressing the dominant, but it's carrying the recessive black and vestigial. Then if you crossed this individual to another recessive individual that has black vestigial wings, you would expect Mendel's rule of independent assortment to produce combinations of wild type versus black versus vestigial versus normal wings in terms of independently assorting those two sets of alleles, right? But what he found was he only got individuals that showed the dominant wild type or individuals that showed both recessive, black and vestigial. And he realized that these factors, these alleles, are actually carried on the same chromosome, and if they're not segregating and recombining, they must be linked. So he came up with this idea of linkage, that certain traits are linked on the same genes. And that's why, I'm sorry, on the same chromosome. Certain genes are linked on the same chromosome. And that's again why Mendel's rule of independent assortment is a rule, because linked genes do not independently assort. They stay together. Okay? However, there's an exception. Sometimes linked genes on the same chromosome can become unlinked, okay? And that's in that process in meiosis called crossing over. Again, crossing over occurs about 10% of the time on our chromosomes. And if it does, what can happen is we can then link or unlink two linked genes so that we now have two genes that are normally not found together appearing together. And this is kind of useful because the chances that two genes will link or become unlinked depends on how close they are to each other. Okay, so for example, when he looked at these linked traits, he found that when he crossed them, he got, of 2,300 individuals, most of them showed that 50-50, showing wild type for both traits or recessive for both traits. However, 206 showed the gray vestigial, meaning unlinking of those traits, and then another 185 showed the black condition with the normal wings. And what he was able to do is figure out that recombination frequency. So if you add these two together, you get 391 divided by the total, 2,300, means that about 17% of the time these genes become unlinked. Okay? Now that's useful because now we can start to look at where these genes are on the chromosome. And a good analogy here is a deck of cards, okay? So I've got two aces here. You recognize them? There's Blanco and Nagin, the two heroes or villains of Katrina. If those two aces are put together, or I put one on the top of the deck and one near the bottom of the deck, and I cut that deck, it's almost 100% chance that those two cards are going to be separated, right? They're going to separate into two piles. However, if I put my two cards very close to each other in the middle of the deck and then cut the deck, it's much more difficult for me to actually separate those two into two piles, right? Because I've got to get right between those two cards. So what that means is based on crossover frequency, we can actually calculate how far two genes are apart. So the larger that crossover frequency occurs, in other words, genes becoming unlinked, the greater distance they are apart on the chromosome. So for example, 17% crossover, what we do is we can say that's equivalent to 17 map units. If we got about a 9.5%, that's much smaller. That means they're much closer together, and crossing over is not going to occur as frequently. That crossover, when it occurs, might occur on another part of the chromosome, but not right in between those two. So in class, we're going to work on some of these crossover frequencies and use that information to determine approximate locations of genes on a chromosome, or relative positions of genes relative to other genes. Okay. So again, Morgan used this information to kind of put together these gene maps, locations of genes on chromosomes. When he found genes that were linked on a chromosome, he could then, based on how frequency crossing over occurred between those genes, determine their relative locations to other genes. And we'll be working on this a little bit more in class. But that's it for this video.